This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 162, recorded December 15th, 2011. Hi, everyone. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
How are you doing there, Alan?
Doing well. It's kind of wet and cold, but that's winter.
Very overcast today.
Yeah.
Gray, dark, getting dark early here. It's 3:30 on the eastern seaboard. Quite dark.
Yeah.
Also joining us today from a place where it's probably not very dark and gloomy, North Central Florida, Rich Condit.
Hi, fellas.
How are you?
I'm good.
Is it sunny there?
It is partly cloudy or partly sunny, depending on your perspective. We got some clouds, we got some blue sky, a little mixture of the two, and it is 76 degrees Fahrenheit. So that's not bad.
All right, very good weather.
Nice.
Yeah, it's
Not
chilly.
bad at all.
It's actually not as cold here as it has been all week. It's warmed up. Let's get up the weather app here. Local weather, New York City, 4 degrees centigrade.
Ooh.
Probably similar in Western Massachusetts.
Yep.
Okay, today we are going to do something we have talked about for a long time, and that is do a Virology 101. Remember those?
Yeah, kind
Yeah.
of.
Alan, I don't think you've done one with us, have you?
I haven't done one. I've listened to a couple of them. They're good listening.
Well, you're taking Dixon's place because he's not here.
Okay, I will ask plenty of
I
questions.
have plenty of questions. And we're going to tackle a topic which follows the previous one, which is DNA-directed RNA synthesis. But before we do that, a couple of reminders. Please take the listener survey at triplemojo.com/TWiV if you haven't already done so. We are approaching our goal, but we're not quite there yet. It would be cool if we got it before the end of the year.
Yeah.
We need to do this.
So please do it. A lot of you are responding. We are at a good number. But not.
And if you listen to TWiV in the car, I'm implanting a subconscious message. The next time you are at a computer, you will go to triplemojo.com/TWiV.
Well done.
Also, pick a session for ASMGM, the American Society for Microbiology General Meeting. We put a link to that in the show notes, and you can suggest a meeting and vote on it as well. Okay, today we're going to talk about DNA-dependent RNA synthesis, which is also known as transcription and is not related to taking an audio program and writing down the words.
Right.
This is making an RNA transcript from a DNA template. And this is a process that all of the DNA viruses that we talk about on TWiV have to do, because you can't make DNA into protein unless you first make a messenger RNA. And viruses do that.
And one of the kind of sort of RNA viruses.
What would that be, Rich?
The retroviruses.
That's right.
Yeah.
Because they make a DNA copy, they stick it in the genome, and there it gets transcribed.
Do we have a separate Virology 101 for retro whatever?
Retro whatever. Yeah, in fact, it's called retro whatever.
Oh, okay, good.
Yeah, we did reverse transcription.
Okay.
And we did RNA synthesis of RNA viruses.
Okay, so this is the last sort of messenger RNA Virology 101.
Exactly.
Right? Okay.
Yeah, and then we move on to other things. I'm not sure if we should do a splicing one or not. That's a pretty cool topic. What do you feel about splicing, Rich?
Well, splicing warms my heart. I think splicing is great stuff. And I suppose, do you have a book chapter on splicing?
Yeah, we do. We do.
Okay. So you're all set to go.
Yeah.
And it's interesting how viruses sort of use the whole splicing trip.
Right.
Now we should probably mention splicing along the way in this episode.
Yes,
too.
I think it is actually. It's it's coming up. But yeah, it's because viruses have their own perversions on splicing, and so it's probably worth an episode. Yeah.
In fact, it was discovered in virus-infected cells.
That
right?
true. That too. And actually, I have my own personal story to tell of the whole splicing thing that we could do. Yeah.
Well, maybe by then we could get some video. So we don't have person video today because I'm not set up to do three-way video yet, but someday we will be. And that's why listeners right now are looking at a famous electron micrograph. You guys don't have this on your screens, but it's an electron micrograph of bacterial DNA in the process of being transcribed. You must have seen this.
It's an old Oscar Miller spread, right?
The DNA is a dark line, and there's just hundreds and hundreds of little RNAs coming off of it.
Right. Looks like a Christmas tree.
Beautiful. It's really beautiful. So that's what we're going to talk about. In fact, a lot of the basic principles that we will talk about today were discovered in virus systems because they're just easier to work with, especially years ago. When RNA polymerase, which we'll talk about in a moment, was first discovered in 1969, the only templates that we could use to study it were total DNA extracted from cells. How crappy that was. So people turned to viruses like SV40, where you could grow virus and purify this circular DNA and use it as a template. So they were early drivers in the study of this process. Now, in cells, we have several different RNA polymerases. Again, these are enzymes that copy DNA into RNA. So there are 3 different eukaryotic RNA polymerases. And I must admit at the outset, this is eukaryotic-centric, this episode.
I can throw in a few prokaryotic tidbits here and there.
That's what you do.
Because I have a sort of a prokaryotic bent in my history, at least.
Yeah, you have it in your blood.
Yeah, you never kind of get rid of that.
So
Rich is bacteremic?
Right. I knew we had you here for a reason, Alan.
He is bacteremic. He has it in his genes also. What would you call that? You can work on it. So we have 3 different eukaryotic RNA polymerases, and they are cleverly called RNA polymerase 1, 2, and 3.
Quite clever.
And these are often referred to as Pol 1, 2, or 3, and often you will hear us using those terms without defining them. And Pol 1 in the cell makes the precursors of ribosomal RNAs. We are now on slide 3, by the way. If you don't want to watch the movie of this Virology 101, we're going to supply a PDF of all the slides so you can go through and look at
for those of you in your cars, here's a subliminal message. Please don't watch the movie while you're driving.
Right.
Really? They can't put the printed out on their steering wheel? And
I know. I don't think we should.
Probably not a good idea. No. That we'd have the National Transportation Safety Board after us.
Okay.
Yeah. Just if you do that, make sure you text us and let us know.
I think we could probably make it understandable without
yes,
figures,
I think
but
we
the
can.
figures will help a lot. So pre-ribosomal RNA Pol 1. Pol 2 is the big guy, makes pre-messenger RNAs and also some of the small RNAs, microRNAs, and that sort of thing. And then Pol 3 tRNAs, ribosomal RNAs, and a few other small RNAs.
Now, why do it this way?
You know, you can't ask why questions.
I know you can't.
Man.
I know you're not supposed to ask why questions. But,
why do you have 3 different polymerases?
Yeah, why not just have subcomponents that allow them to do these different things?
That's really an interesting question. The jobs are really very strictly compartmentalized. So Pol 1 does ribosomal RNA, and you need a lot of that. Okay, and so
Right.
I would assume. Okay, so these are all multi-subunit enzymes.
Mm-hmm.
I think they have a couple of subunits in common. Is that right? The 2 large
That's right.
guys.
That's
I think
correct.
that's-
Where most of the action is, at least most of the polymerization action. And by the way, those 2 large subunits they're homologous to the 2 large subunits of the bacterial enzyme. So this is a theme that is very well conserved throughout biology. And then they have a bunch of ancillary subunits that I would assume are specialized for these specialized jobs. So if all you're going to do is make ribosomal RNA, as you'll see as we go through this, there has to be lots of variability in what some of these enzymes can do, especially the Pol2, the messenger RNA synthesizing enzyme. So if you're going to have, you can make a lot of ribosomal RNA, it probably makes sense to have an enzyme that's specialized just to do that. Likewise for the small RNAs, and the Pol2 that makes messenger RNA has to interact with a bunch of different factors and that kind of stuff, so it probably makes sense to have a specialized enzyme to do that. I don't know if that's making any sense, but that's kind of sort of where my head's at on Alan's question.
According to the textbook, the evolution of RNA polymerases with distinct responsibilities appears to be a device for maximizing opportunities for regulation of mRNA synthesis while maintaining a constant and abundant supply of RNAs essential for cellular metabolism.
Yeah, that's what I said, right?
Yeah, that's exactly what Rich said.
Exactly.
Right, so you always need your ribosomal RNAs and your tRNAs, but you may be turning on and turning off different messenger RNAs at different times.
Right. So the way to ask the question would be, for what reason did these three evolve? And that way we can't say, you can't ask a why question.
Yes.
I like the why questions. I don't care what anybody says. I like the why questions.
So Pol1, as far as we know, there are no viruses that utilize Pol1 or need it for their replication, but they do use Pol2, of course, to make messenger RNAs. And then some viruses, adenovirus is one of them, Epstein-Barr virus, utilizes Pol3 to make smaller RNAs.
And these are complex enzymes, okay? Like I said, 2 large subunits and a gaggle of small unit subunits. It's like 10 to 12 subunits altogether.
12 to 16 subunits.
12 to 16. Yeah, like I said.
It's exactly what Rich said.
They're big. They're pretty big. And
as opposed to coli, which is 4 subunits.
Right. And some of these big enzyme structures have been solved. The 10 subunit form of yeast RNA Pol2 has been solved. That's amazing stuff. I bet it's Stites is one involved in that.
Roger Kornberg.
Roger Kornberg.
Roger Kornberg did the. I don't know that Tom Stites did any of the mammalian RNA polymerases. Roger Kornberg is responsible for most of that stuff.
Okay. All right, moving on to the next slide. Slide number 4. Now, viruses, all the DNA and retroviruses that we're going to talk about today, have different requirements for transcription. So, retroviruses, for example, whose DNA copies go into the cell, they just mostly utilize the cellular transcriptional machinery. Right? They just stick themselves in there, and the cell, as it's transcribing cellular genes, it transcribes retroviral genes. And other viruses add to the complex. They add their proteins. Some viruses use all the host components, as do the retroviruses, and they add a protein. So some of the bacteriophages, T3 and T7, do that. Parvoviruses. Some of the more complex retroviruses, like HIV, they add their own proteins, papillomaviruses and polyomaviruses. And then the bigger DNA viruses, adenoviruses, herpes viruses, phage T4, they make a bunch of viral proteins, and that's important for the regulation of transcriptional patterns, as we'll see later. And then we have the pox viruses.
I've decided to go it alone.
Boy, I guess that's why you work on them, right, Rich?
The heck with a host. No, I work on pox viruses because they remind me of phage T7. and that's the truth.
Really?
Okay, yeah. They're very lytic. You know, they shut off host the heck with the host. They shut it right down and do their own thing, and you get a very nicely scheduled progression of gene expression, and you just don't have to worry about the cell. The cells, I mean, that's my perspective, right?
Right.
When you get into host immune response and stuff, you're talking about something different. But with the talking about the nuts and bolts of gene expression, I like the fact that they're independent, so you don't have to worry about the bureaucracy of the host cell.
Now,
so,
there are different advantages and disadvantages to these strategies, though, from the virus's perspective, right?
Well, if you want to bring in your own proteins, you need to have bigger genomes. Yeah, that's one.
Right. And also, you run a higher risk of tripping off some kind of host immune response.
Mm-hmm.
Whereas if you don't bring in your own tools, then you're entirely reliant on the host, and you may not like its gene regulation patterns.
Yeah, the RNA viruses do it all themselves pretty much because there are no RNA-dependent RNA polymerases in cells, so it seems to work for them.
And I would say, in terms of these guys that use a combination of host and viral proteins, you've kind of said this on this slide, but I want to emphasize that there's two fundamental strategies. One is that the virus comes in and uses the host transcription apparatus to transcribe part of the genome into a set of early genes, and one or more of those gene products is a new virus-specific RNA polymerase, which has actual transcription activity and can transcribe the rest of the genome. And that includes both the phages T7 and T3 and some others, and a couple of eukaryotic viruses. The baculoviruses do this. They make their own RNA polymerase, and the iridoviruses do that. They make their own RNA polymerase. They're more complex structures. The T7 and T3 bacteriophages make RNA polymerases that are a single subunit, which is why they're so useful in biotechnology. The baculoviruses and iridoviruses make more complex enzymes. The baculoviruses make a 4-subunit enzyme that, once again, has 2 large subunits that are homologous to the coli and mammalian subunits and a couple of smaller subunits. And then the other viruses that use a combination of host and viral proteins, rather than making their own polymerase, they make factors that in one fashion or another tweak the host's apparatus to direct its attention towards viral genes and stimulate viral transcription. But you're still using the host RNA polymerase, usually Pol2.
Right. So you can use the host polymerase to just as a bootloader to boot up your own system, and then you do your own transcription.
Right.
Or you can continue to rely on the host and just guide it with your own regulatory proteins.
And we'll look at some examples of that a bit later. All right, let's take an overview of this whole process of transcription on the next slide.
Slide 5 we're at.
Slide 5. Yep. Numbers in the lower right-hand corner, courtesy of Rich Condit. Now, these viruses, for the most part, and there are exceptions, as Rich will tell us, have to get into the nucleus. If you're depending largely on host RNA polymerase and host proteins, associated proteins, the DNA has to get in the nucleus. Of course, one of the exceptions are the pox viruses, which do this in the cytoplasm, and there are other viruses as well. So whatever your virus is, an SV40, a parvo, an adeno, a herpes virus, the DNA has to get into the nucleus. So in this slide, we're showing that. You can see the nuclear membrane with pores. At the very top, there's a double-stranded DNA, and the red arrow is where transcription begins. That is the initiation of RNA synthesis, in this case, mRNA. And these mRNAs are largely made as precursors, but not always. Some viruses don't make precursors. In this case, we're showing mRNA precursors that undergo splicing to remove introns. So the parts that are removed are called introns, and the parts that are kept are called exons. No relation to the gas company.
And it's only 1X.
Right. And these mRNAs also have a unique chemical structure at the 5' end called the cap, which we talked about in our translation. No, we didn't do translation yet, which we will talk about in translation. It's very important for translation. It's important for export mRNA stability. There's a very unusual 5' to 5' linkage of the terminal G residue. Normally, the bases are linked in a 5' to 3' direction. Cap is reversed.
And viruses focus a lot of attention on that cap. There's lots of virus tricks with the cap.
Right. Many viruses use host enzymes to put the cap on. Vaccinia makes its own to do that.
Right, because if the RNA is not capped, then the cell is not going to process it in the normal way.
Right.
And when you think about it, if Vaccinia is going to do all of this in the cytoplasm, it has to supply the capping enzyme, which is a nuclear enzyme.
Bingo.
So the mature mRNA, it's capped, it's spliced, it's polyadenylated as well at the 3' end. There's a stretch of poly-A's. We'll talk about how those are added. Those get exported through the nuclear pore complex into the cytoplasm, where they can then be translated. That's the overview. Anything to add, gentlemen?
No, well, we touched on splicing, and I guess that'll be a whole other topic, because there's a whole lot to that. But the reason for splicing in eukaryotes is that our genes are split up into these little segments, these exons that code for the actual protein. And then in between those, we have varying lengths of stuff, which some people call junk, and some people say is useful, and some of it is useful, and some of it we don't know what it does. But those are the introns, the sections that are of the DNA that are not coding for proteins. So you get initially this full-length transcript, has all the exons and the introns, and then that gets spliced so that it's just exon 1, exon 2, and so on.
And this allows a modular arrangement of genetic information.
Right. You can take pieces of different proteins and move them around.
Right. So it gives a lot of flexibility.
All right. On the next slide, number 6, these are the steps in transcription that we're going to talk about today. First, promoter recognition. We'll tell you what a promoter is in a moment.
And it has nothing to do with Don King.
Pre-initiation complex formation. Initiation, elongation, and then termination. So those are the individual steps. We thought it would be nice to have them all listed out here for
and
you.
We talk about this stuff all the time. I'm glad we're finally getting around to defining it.
Yeah, we do. We do throw these terms around, and they're not immediately obvious to what they are. All right, so let's look at promoter. Let's look at the beginning part, which comprises promoter recognition and pre-initiation complex initiation. And we'll go to slide 7, which shows a generic control region of a DNA molecule. And this is an area that's important for the initiation of RNA transcripts. So this whole process of transcription begins in this area, which is called the transcriptional control region. And this is a pretty complicated region. It can be quite large, as you can see by the numbers at the top. The numbers at the top tell you the base pairs involved. So all the way at the right, you see the red arrow, and that is where transcription begins.
So the gene would be to the right off the slide.
That's right.
We're just talking about where it starts.
And it begins in that yellow box. It's called an initiator sequence, and it's a sequence of bases that are defined to be important for initiation. And just to the left of that, within 20 or 35 base pairs, is the TATA sequence, TATA. A TA-rich sequence, which forms part of what's called the core promoter.
That's, for those of you with your minds in the gutter, that's TATA is the actual sequence. That's why it's called a TATA sequence. We didn't make that up.
Some people call it a TATA box.
Yes.
Originally the Goldberg-Hogness box.
That's right. And this is bound by a protein called the TATA box binding protein, TBP. Also has another name which will come up. What is it, Rich? TF2D?
Oh, geez. Now you're going to get me into factor
it's
comics.
All right. Don't worry about it.
No way.
It'll come up later. TATA box binding protein. Anyway, not all promoters have TATA sequences, but many of them do. Then moving to the left, the promoter continues. It's comprised of what are called local regulatory sequences. Basically, if you mess with them, you mess up transcription. For many promoters, that core promoter is enough to get transcription. And then everything else adds regulation, which are important in various situations in development and growth and so forth.
Response to various environmental conditions, etc.
Right.
You got to turn genes on and off at different times and modulate them at different levels. And so you need to be able to have things that can regulate the transcription, and those local regulatory sequences are part of that. The core promoter is just polymerase binds, transcribes. The regulatory sequences modulate that reaction.
And then we have some very distant regulatory sequences. They're called either enhancers or silencers, depending on whether they stimulate or inhibit transcription. And you can see they can be 100 or over 10,000 bases away. And we'll talk about how those work.
Right. Now, the purpose of this whole transcriptional control region, the core promoter, the regulatory sequences, the enhancers, the silencers, is there's a whole lot of DNA there in the nucleus, and something needs to tell the polymerase complex where to start and when to start.
Right.
Mm-hmm. And that's what all this stuff does. The core promoter tells it where to start, and then all the regulatory sequences tell it, are involved in saying, okay, transcribe that, but not this.
Right. And the when, that's a complex when. That's a
Yes.
when in response to environmental cues or developmental cues, and it's also when in the sense of how frequently.
Yes.
You want a whole bunch of this or just a little bit?
Now, every transcriptional unit has some kind of variation of this area in front of it, right?
Mm-hmm.
Right.
Because every gene has to be regulated. And so depending on where the gene is and what it's doing, it can be very much like this. Now, the other thing we should mention is that DNA isn't just floating around in a cell in the nucleus. It's all wrapped up in eukaryotic cells. And it's wrapped up around histones, and it forms little what are called nucleosomes. And it's pretty tightly wrapped around these proteins. And so another level of regulation is to loosen up that nucleosome structure and allow the proteins to get in. And
now, Pol 1 and Pol 3 that are mostly transcribing housekeeping type stuff, do they tend to bind to less complex regulatory sequences?
Yeah, there are also factors involved for those polymerases, but
I think it is, however, true that it is less complex.
Yeah, it's less complex.
You don't have all this.
You generally want those on.
Yep.
Right. And this raises another point as well, and that is that viruses generally don't have to modulate their transcription during infection or from infection to infection. In any given infection, except for viruses that maybe have a latent phase, you pretty much want to transcribe all the genes in the same on the same sort of schedule and to the same sort of amount. So the regulation that's going to go on there is primarily redirecting the attention of the host polymerase to the viral genome to do this on the same schedule. But you'll see a lot of regulatory sequences associated with that, but they don't have to modulate over time or in response to various conditions the transcription of a given gene. So, for example, in pox viruses, where the transcription is entirely self-contained, there's none of this regulatory stuff. There's basically a core promoter sequence that the virus polymerase responds to, and there's no other regulation overlying that. That's one of the advantages of going it alone. You don't have to deal with all of this complicated stuff.
Well, yeah, that's cool. You just blast out the mRNAs.
Right, just do it.
All right, next one is slide 8. This is an overview of the initiation process.
This is good. I like this.
So we start at the top with our double-stranded DNA. The yellow box is the control region that we've just been talking about, and the red arrow going to the right is the site of mRNA initiation. And the RNA polymerase is shown here, RNA polymerase 2. It's a rather large complex. And then there are all the initiation proteins that interact with various sequences in the core promoter. And they come together with the polymerase. And you can see one of them is TBP. That's the TATA binding protein. And
that sort of nucleates the whole thing,
yeah,
right?
It brings it to the core region by interaction with the TATA box.
And a common theme for all of these guys when you have an initiator protein is that it's going to have to interact with DNA that's upstream from the start site, and also either directly or indirectly through other proteins interact with the RNA polymerase. Okay, I mean it's obvious, but it's worth pointing out that going to bind one of these accessory proteins has to bind DNA and bind the polymerase, and in the process help the polymerase find the right site to start.
So the first complex formed, the polymerase and all these initiation proteins, it's called a closed complex because the DNA is double-stranded still. It needs to be melted and opened up so the polymerase can access the template strand, right? Because it's going to copy just one strand not both. And so to open up this complex to form what's called the open initiation complex, you need energy to melt the DNA. You need a helicase. What is the enzyme called, Rich, that would open this up? Or the activity?
It'll be something with a helicase activity, yes.
Yeah. So now it melts the double-stranded DNA. ATP is needed for that, and you have an open initiation complex. You can start making RNA transcripts. So that's what's called the initial transcribing complex. Now, the polymerase wants to begin moving down the DNA and copying it, but it's still attached to these initiation proteins of various sorts. Those are the orange ones in this figure. So until it can detach, it tends to make a lot of short RNAs. These are called abortive transcripts. And then at some point it breaks away and starts to go down the DNA and makes what's called the nascent RNA transcript. That
breakaway they call promoter escape.
That's great.
And the abortive transcripts, I don't know if there's I don't know if there's any function to those. I'd have to think about that. I'm thinking about actually some coupled transcription translation stuff that goes on in coli where the abortive transcripts may play a role, but I won't worry about that. At any rate, it seems like a wasteful process, but it's very common. It's common to both eukaryotes and prokaryotes. And by the way, all of the fundamentals of this are the same in bacteria, and in fact were worked out initially in bacteria.
Mm-hmm. Right.
Basically the same thing, just fewer proteins, less complex.
Okay, so the initiation proteins call in their buddy RNA Pol2 and say, hey, this is the place to hang out. So it then is stuck to the initiator there, and it tries to leave, but they're all still stuck together.
Right.
So in trying to leave, it starts to transcribe, hey, come back, have another one, and it just keeps stuttering along there until some energy gets applied that separates these two, and then
boom,
off goes the polymerase.
Right. All right, moving on to number 9. This is a cool picture I wanted to show because it is a structure of the TATA binding protein bound to the TATA sequence. So the protein is in the green ribbons, and the DNA is in blue below. So we have the structure of that. The interaction of TBP with the TATA sequence is thought to bend the DNA. So on the right is a diagram of that. Here's again the RNA Pol2 with all the initiation proteins. And you can see now we have these wonderful names, TF2A, TF2B, which are named because they were fractions coming off of a column. Mm-hmm. And that's it. The TBB is shown there binding the TATA, and it's bending the DNA. And it's not really understood why this happens, as far as I understand. But one idea is that this bending facilitates the interactions among all these regulatory proteins.
And once again, keep in mind that this is all happening on a nucleosome. So the bending may have a different sort of context there.
And also bear in mind, these things don't always need reasons. It's possible that the bending is a byproduct of some other structural feature of the TBP that's just necessary to get this whole complex together. Or it may have some purpose.
All right. On the next slide, number 10, are just some examples of these control regions in viral genomes. So far, we've looked at sort of a generic control region promoter. Could be from a cellular gene, but now these are the way these things are arranged in adenoviruses, where you have an adenovirus major late promoter. So AD has a number of different promoters to drive transcription. And you can see the relationship of the initiator and the TATA box. Below it is the SV40 early promoter. The TATA box in this one has multiple initiation sites. You can see by the 3 arrows. And also the adenovirus 4A2 promoter, where you can see there's no TATA box. And then finally the SV40 late promoter, where there are 3 different overlapping initiation sites.
Now, DNA viruses commonly have these early, middle, late types of promoters. Why do these DNA viruses care so much when things get transcribed?
Well, there's a the way I see it, there's a strategy, an organizational strategy to the infection where you go in, and the first thing you have to do is take control of the host, because the host is gonna really try and fight this off. Okay, so you devote all of your attention early on to doing two things. One, if you can, launch some immune countermeasures. All right, and stuff all of the cellular and organismal mechanisms that are going to try and get rid of you. And two, establish transcriptional dominance over the host, which means either making a new polymerase or some new factors or something that are going to tweak the cellular machinery. And at the same time, actually also set up DNA replication. So once that's done, you're done with that. Now you can enter a new phase. That new phase is DNA replication, and you devote all of your attention to that. Once that's done, now you can get on with assembly. And that involves a new transcriptional class. That means making all the structural proteins and putting it together with the genome. Now, it's hard for me to think why it has to. I mean, it seems logical to me to do it in a cascade like that. Why you couldn't do it all at once, I don't know. But
right, I guess it's a more efficient use of the available cellular resources. So it's whatever the top priority is for the virus at that point in its life cycle is what's getting transcribed.
Well, certainly there has to be. It seems to me at least two phases from the point of view that when you go into the cell, if you're going to use the host transcriptional machinery, when you go into the cell, you have to use what's there, okay?
Right.
And then if you're gonna modify that to really stimulate viral gene expression, preferentially to host gene expression, that means that some of your initially transcribed genes have to be these new factors that you're going to use to modify the transcriptional apparatus. So right there, you've defined two phases, okay, just operationally.
Right.
And whether or not it's convenient, I guess it is more efficient to segregate out DNA replication and assembly as well.
We're going to talk.
And I guess also you have initially a very small number of genomes, maybe only one, in the cell, and you don't want to be taking up its available space by transcribing a bunch of capsid genes.
Exactly.
When you don't need them until you've gotten to that point.
That's a major driver. We'll get back to this a bit later. We're going to talk about that. The next slide, number 11, shows you some of the, again, viral DNA genomes and this transcriptional control region. You can see the red arrows on the right, the initiation sites for synthesis of RNAs. And these are just some of the specific proteins. These are cellular proteins that interact with adenovirus or SV40 promoters of various sorts. And you can see they have weird names, CPL, USF, SPL. And I will quote from the textbook, the nomenclature applied to these regulatory proteins presents serious difficulties for both writer and reader, for it is unsystematic and idiosyncratic.
Bingo.
That is Jane Flint writing all the way.
I was going to actually, did Jane write this chapter? Do you guys split this up by chapter?
Yeah. This is Jane's chapter, yeah.
Okay.
You can tell this is her writing. So these are just some of the regulatory proteins that interact with these sequences around promoters and are essential for regulating when stuff is going to be made. All right, the next one is 12, and this is a diagram of how we think enhancers work. Remember, the enhancers are these distal sequences. It can be over 10,000 nucleotides away. That, in the case of enhancers, stimulate initiation of transcription. So here you can see an initiation complex formed in the promoter region. There's our pink RNA polymerase, all the initiation proteins. And then the enhancer is far away, but it's folded so that the DNA of the enhancer sequence is near the initiation site. The enhancer's in orange. You can see there are enhancer binding proteins that are linking that sequence to the initiation complex.
Right. And so the shortest distance across 10 kilobases of DNA is a fold.
That's right.
Right. And if I'm not mistaken, there have even been like electron micrographs where you can show this thing actually forming.
That's right.
At least in a test tube.
Right.
All right, moving on to number 13. This is a diagram of what many of these regulatory proteins, these are all the various proteins that are binding these sequences in the promoter control region. They tend to be modular. They have shown here 3 distinct domains. They're called the DNA binding domain of the protein. This is, of course, the part that binds DNA directly. There's another part that allows more than one copy of the protein to interact. That's called the dimer formation domain. And finally, there's the activation domain. That's the part that can stimulate the transcription of RNA. So these are called modular because you can actually mix them around among different proteins.
And typically that activation domain will interact with another protein or directly with the RNA polymerase. So there's the glue there. You're gluing some other protein like the polymerase or some other protein as part of the complex to the DNA. You're guiding another protein to a specific DNA sequence using the combination of the DNA binding and the activation domain.
Right. Now, how do these proteins look like on DNA? That's shown in the next slide. Here's an example of a particular protein binding to DNA. As you see at the bottom is the helical DNA in orange. And then the protein, there's 2 copies. It's a dimer going vertically. And these are 2 very specific proteins called Jun and Phos, which make up a very famous transcription protein. And you can see the protein binds the DNA, and then there's a part called the leucine zipper, which is the part that allows this particular protein to dimerize. So that's
cool.
Just one example of how these work. It's pretty cool.
And I just as an aside, I know because this interests me, I noticed in the text it says that those leucine zipper domains are unstructured if the thing is not bound to DNA. So they're just kind of flopping around.
Yeah.
It's not bound to DNA, and when it binds to DNA, they form this structure. And there's a lot of proteins, regulatory proteins, that are like that, contain a significant amount of the protein that's not necessarily very structured, but when it undergoes a certain interaction, it will now assume a structure that's appropriate for whatever that interaction is.
Right, and it has to do it as a dimer.
Right.
So if you have one of these proteins but not the other, it can't do this. And if you have both of these proteins together but they're not stuck to DNA, they don't do anything.
Remember that we've eliminated all the other proteins in this complex. Everything else is gone, just to illustrate this here.
Right.
And it's not all these proteins actually bind DNA. Some of them can activate transcription in other ways. We don't have time to get into that. All right, the next slide, number 15, really gets back to this issue we touched on, which is that why do viral transcription patterns occur in waves? And these are 2 different ways that viruses can regulate transcription. And, as we said, as a virus infects a cell, initially there's just a genome, and you very quickly have to ramp up production of transcripts. You have to first work, as Rich said, with what's present, but then many viruses modify the transcriptional apparatus, make their own transcription proteins and so forth, so that they can favor their own synthesis. So here are 2 different strategies. At the top, there's one called a positive autoregulatory loop. So the viral genome gets into a cell, a gene is transcribed. The translation product of the mRNA is a transactivating protein. That is a protein that increases the transcription of that particular gene. So it's amplifying itself by a positive loop.
Right, it's a feedback.
And this happens in cells infected with HIV-1, for example. And then the one on the bottom, shown as B, you have, again, a viral genome. The initial transcript gives rise to a protein called X. And then, and of course, that transcription depends on host machinery. Then protein X is actually a transcriptional regulatory protein, and it can help turn on the transcription of later genes, ones that you don't need early on. And
that's where you get this scheduled early, intermediate, late cascade of transcription.
Right. Exactly.
Right. So gene Y can't be transcribed until protein X exists, and protein X can't exist until gene X gets transcribed.
Right. And we don't want Y at the beginning of infection, as Alan said. If it's a capsid protein, you don't want it till later on.
Right.
So you have this temporal regulation. All right, the next slide is a table. It's slide 16. Just show you some of these viral proteins now. We've talked a lot so far about cellular transcriptional regulators with all those funny names. Here are some that are encoded in viral genomes, and they have funny names too. Adenovirus, the 4A2 protein, herpes virus, ICP4, Epstein-Barr virus, ZTA, then there's a papillomavirus, E2, and then vaccinia virus, VETF.
Right, and the way they get these funny names is somebody working on adenovirus came up with names for all the genes initially, and then it turns out that 4A2 happens to have this property, and separately somebody working on herpes viruses had the ICP proteins named, and then it turns out that this is the function. So we got at their functions in different ways, so you get very different names for proteins that have similar properties.
And again, these have specific functions. So, for example, the 4A2 protein of adenovirus is important for stimulating a late promoter. So without this protein, which is made early, the late promoters don't work. And again, it contributes to this temporal regulation of transcripts. All right.
So these all fall into the class of factors that actually bind DNA and do something.
That's right. They bind DNA. Right. Now, Rich, this VETF, do you know something about that?
Yeah, I know a thing or two about it.
So
viral early transcription factor.
So the reason the virus has brought in its own RNA polymerase, right? Mm-hmm. And it can't access any cellular transcription protein, so it has to make its own as well, right?
Right. And so it packages the RNA polymerase, and it actually undergoes this cascade of transcription: early, intermediate, and late, for all the reasons that we've already discussed. And it packages the RNA polymerase in the virion, and it has to have a way of selecting out and transcribing just the early subset of promoters early during infection. And the way it does that is to package also this viral early transcription factor, which is a DNA binding protein, binds to DNA and binds the polymerase, as you'll show in a cartoon coming up. And then during infection, one of the early gene products is a new transcription factor that activates intermediate genes. One of the intermediate gene products is a new transcription factor that activates late genes. Those probably somewhere along the line have DNA binding components in them as well, but the mechanism of those hasn't been as well worked out. The only one that we've proven is a DNA binding protein is this VETF.
Right. So vaccinia does away with the cellular transcription system, and therefore has to reinvent all the pretty much the same machinery to accomplish its
right
goal.
But it's much simpler. Okay. Right. The polymerase is the polymerase is a little simpler. We're talking 8 or 9 subunits, depending on whether it's early or late, and the factors, rather than a dozen or 2 dozen subunits, you're talking about, in the case of VETF, it's just 2 subunits. And the others have 2 or 3 subunits. And like I said, you don't need all of the really fancy regulation, so it can be pretty straightforward, pretty simple. That's the reason for studying it, by the way. Okay, is that it's not nearly so complicated, right? So you have a chance of understanding it.
I presume viruses do a similar thing because they replicate in the cytoplasm.
I presume.
And all these large DNA, nuclear cytoplasmic viruses do similar?
I presume so. They are not as well worked out. Certainly the ASV viruses. Now, I guess in that class, we should include, as I was talking about before, the baculoviruses and the iridoviruses.
Right.
And they're a little different because they start off using the host polymerase and then make their own new polymerase. And actually, the iridoviruses are really strange because they start off in the nucleus and transcribe early genes using a host RNA polymerase, and one of the early gene products is an RNA polymerase, and then, in fact, they move the whole thing out to the cytoplasm and finish off DNA replication in the cytoplasm and do the late transcription in the cytoplasm. The baculoviruses, I think, do the whole thing in the nucleus, even though they're using a viral RNA polymerase for the late phases. Another large DNA virus that I don't know if it's included in the nucleocytoplasmic viruses are the chlorella viruses. Infect blue-green algae, and these are large double-stranded DNA viruses. And they got enough room in their genome, so they do stuff like encode tRNAs and restriction enzymes, right?
Yeah. Cool.
Okay. But they don't have any RNA polymerase subunits. So they're probably relying entirely on the host. I believe that the Mimi viruses and et cetera, the genome sequences show transcriptional stuff like capping enzyme, RNA polymerase, that kind of stuff. So presumably they are self-contained, and as you already said, they replicate in the cytoplasm. So that would mandate that. Yeah. But nobody's done sort of the biochemistry on those.
All right, the next slide, 17, is a summary of three different viral strategies for these kind of cascade regulation steps of regulation of transcription. The top one is SV40. It's a simple one. SV40 has early and late transcription patterns. So when the virus infects a cell, the first messages that are made encode this protein called large T antigen, LT. And this is then required, and that's early gene expression. And that protein is then required for activation of the late promoters that would then make mRNAs that encode capsid proteins.
And at the same time shuts down early gene expression.
Correct. And stimulates viral DNA synthesis, which is then you got viral DNA and capsid protein, so it makes perfect sense. So that's it.
So that's a really multifunctional protein, and that makes sense because this is a small virus. On the top, you're saying, actually, you got these in order of size. SV40 on the top, you're talking 5 kilobases. That's only worth about 5 genes. Adenovirus, you're talking about 40 kilobases. I think that's about 40 genes. HSV, you're talking about 100 kb. That's 100 genes. So these are in increasing complexity as you go down this slide.
Right. So the adenovirus, which is in the middle, when its first messages are called immediate early messages. And these encode a protein called E1A, which is required for the production of early transcripts. And then one of the early transcripts encodes an E2 protein, which then is required for the production of late transcripts. So 3 different phases and a cascade of proteins required for each transcript. And then finally, herpes, which is the biggest. Herpes also has immediate, early, and late, but in this case, the immediate early genes can't be transcribed unless the virus brings in the particle, a regulatory protein called VP16. So that's physically brought in with the particle. It allows the production of immediate early genes, which include 2 regulatory proteins, ICP4 and ICP0. 0 is required for the production of early transcripts. And among those are replication proteins, which drive DNA synthesis, of course. And those, along with ICP0, are needed for entry into the late phase of transcription. So herpes is the most complex. It turns out that of all the 80 or so genes of herpes, each one has its own regulatory program. It's really amazing. So AD has a couple of promoters that are regulated, but herpes, for each gene, it's independently regulated.
Its own promoter.
Yeah. Now, this VP16 protein is very interesting, and so I thought I would throw in the next slide, number 18, which is 2 models for how VP16 works. And why this is interesting is it provides some more general principles about transcription and its regulation. So it
provides a nice model for exactly how a virus can tweak the cellular machinery to make it work differently.
Yeah, so the goal here is the virus wants to go in and start getting its genes efficiently transcribed so it can start taking over the cell. And so VP16, the idea here is on the top, remember we have our closed initiation complex with the RNA polymerase joining initiation proteins. And this is not a terribly efficient process, and one of the problems is this protein TF2B, which exists all folded up. And in order to bind the RNA Pol initiation complex and participate in initiation, it has to unfold. And what VP16 might do is helps this protein unfold. So with VP16 around, you get very efficient formation of initiation complex on viral DNAs. and that's thought to stimulate transcription. The model at the bottom is really cool because now we show how the DNA actually really looks like, both cellular and viral DNA. It's wrapped around these assemblies of histones called nucleosomes. And when you're wrapped around a nucleosome, it's very hard for the polymerase and for the associated proteins to access the TATA box and the initiation site. And so one idea is that VP16 helps to loosen up the DNA that's coiled around the nucleosome and therefore allow access of VP16 and all the initiation proteins as well as POL2.
Cool.
I think that's pretty cool.
Mm-hmm. That is neat.
This whole wrapping and unwrapping involves a lot of modification of histone proteins, which I think eventually we'll talk about one day. I'm sure we'll have a paper that addresses it. It's a really hot area of research at the moment.
Now, herpes viruses, I would assume that they would develop a complicated regulatory program like this, at least in part because they have a latent phase, right?
Mm-hmm.
So they need to be able to hide out in the cell and not transcribe their genes the way they would during the lytic phase and just be latent.
Yeah. In fact, they have to have a way to shut everything down.
Right. And some herpes viruses have a whole bank of genes that are just devoted to regulating that balance between lytic and latent infection. And some of this, the current thinking is that some of that regulation has to do with the histone modifications that govern the nucleosome configuration of the genome. So it's complicated.
Yep.
Right.
All right. Next slide, number 19, is an example of how adenovirus proteins can modify transcription sites. So in a similar way to the VP16 of herpes viruses. There are two examples shown here involving an adenovirus protein called E1A. Remember, E1A is one of the transcriptional regulatory proteins specified by adenovirus. And at the top, you can see that the E1A protein is shown as a blue line. It's a zinc-binding protein that helps maintain a certain structure of the protein, and it interacts with a variety of cellular transcriptional regulatory proteins. They're shown here as MED23 and ATF2, and helps to recruit these proteins to the initiation complex. So this allows, again, as soon as the virus infects, to make their own genes preferentially transcribed. Now, the bottom is a very interesting scenario that we will again come to in the future when we talk about transformation. And this is the effect of the E1A protein on cellular genes that have what's called an E2F site in them. And this E2F site is just one of these regulatory sequences in the promoter region. The E2F promoters drive genes that are involved in cell division. And so it's very important that they be regulated. In fact, in a cell, there is a complex of proteins, they're called DP1, E2F, and RB, that bind these promoters, and only under certain conditions when the cells have to divide are they removed. So these are repressor proteins. The RB protein is an abbreviation for retinoblastoma protein, and it was identified in children with the tumor retinoblastoma. They have mutations in the gene encoding this protein that make it not regulate transcription properly, and therefore cells divide uncontrollably. Now, the adenovirus genome, for replication of it, needs to have E2F activated because it needs some of the proteins that are made. So the E1A protein basically bumps off the retinoblastoma protein and activates these E2F containing promoters. So the virus needs that for replication, but the result is it can sometimes lead to cellular transformation.
And so that's a general problem that that viruses can cause when they're trying to get DNA replication in a cell, right? That's why we have viral oncogenes.
That's right.
It's interesting that well, first of all, I remember this story developing, and it was absolutely fascinating. It was wonderful work. Joe Nevin's responsible for a large fraction of it. And the real puzzle initially was that it was known that the E1A protein was a transactivator. Okay, that is, that in the absence of E1A, you did not turn on the E2 genes in adenovirus. By the way, the E2 genes encode all of the DNA replication proteins,
right,
so that's phase 2 of the infection. And yet, at the time, really most of the thinking was that all of these transactivators should be of the DNA binding type. People hadn't thought about this indirect stuff very much, okay? So, and everybody tried to show that E1A bound DNA and all that kind of stuff, and it didn't, okay? And then it turned out that, in fact, the action of E1A is indirect. It does what's shown here, which is to compromise the function of a repressor protein, so it can actually activate a gene without binding the DNA. I wonder how many people who study transcription and work on, for example, or for transformation for that matter, and work on the cellular protein E2F, I wonder how many of them understand that E2 stands for the E2 region of adenovirus.
Yeah, that was discovered initially, right? Yeah. It turns out that both the cell and the virus have similar promoter control regions. That's what's going on here. So
it's interesting to me as well that E1A is killing two birds with one stone here. Okay, it's turning on its own DNA replication stuff by knocking RB off, and at the same time stimulating cell cycling, or stimulating synthesis of cellular DNA replication enzymes, which will indirectly impact on virus replication by providing more replication apparatus.
Absolutely. Yep. All right, we have two more. The next one I'm going to give to Rich, because
this is what we've discussed already. This is how the initiation factor VETF works for stimulating early viral transcription in pox viruses, and quite simply, it's VETF is a heterodimer, and it binds to a sequence upstream from early promoters. The viral RNA polymerase actually comes in two forms. There's a core 8 subunits that exist in both forms, and then there's a 9th subunit called RAP94. VETF, by the way, is viral early transcription factor. I think I already said that. RAP94 means RNA polymerase associated protein that is 94 kilodaltons molecular weight. So that thing, it's depicted as a separate protein, but in fact, it's really a subunit of the viral RNA polymerase that is used early during infection. And it confers specificity for early promoters by virtue of the fact that it binds to VETF, and so that binding interaction recruits the RNA polymerase to the early promoters, and now it can initiate transcription. And actually, Vincent, I think the actual proof that RAP94 binds to VETF probably happened after the. I wonder if it happened after the last edition of the book. If this was, this could easily be presumed, but I wonder if it was known that interaction actually took place. At any rate, it does. So there you go.
Yeah, it seems to. According to the legend, it sounds like it was known.
So here's an interesting thing, and that is that RAP94 also is part of the termination complex. Early genes are terminated in transcription is terminated by a mechanism different than late genes, and that subunit RAP94 interacts with termination factors so that the polymerase knows what the termination sequence is for the early gene as well. So it's important for both initiation and termination.
All right, our last slide is just to tell you what happens at the end of all of this when transcription ends and the mRNAs are polyadenylated. So we're starting at the top. That is an RNA transcript being produced by RNA polymerase II. And in eukaryotic cells and most viruses, the transcript goes beyond what's called the poly-A addition site, and you'll see why that is in a moment. It terminates farther downstream. The signal to add poly-A to a messenger RNA is a sequence called the poly A addition site. It's shown here as AAUAAA. And there is a protein in cells called CPSF, which cleaves the mRNA just downstream of that site. And then a variety of other proteins shown on this slide, including an enzyme called poly A polymerase, then come in and add a sequence of A's to the 3' end that was generated by the cleavage by that CPSF protein. And that's called polyadenylation. And eventually you add about 200 A residues to the 3' end, and you now have your mature messenger RNA that's ready to be exported into the cytoplasm and be translated into protein. And now I understand, Rich, that vaccinia does this totally differently, right?
Well, so yes, as far as we know, for neither the early nor the late genes, well, late genes is a little ambiguous, but certainly for early genes, there seems to be no cleavage reaction, okay? And the virus makes its own poly A polymerase, which will basically polyadenylate almost any available 3' end, but probably does so in a concerted fashion with the polymerase in the virion. With intermediate and late genes, the presumption has been that terminated RNAs are just polyadenylated. It's known that that's done by the same virus-coded poly A polymerase, but there's some suspicion that there may be a cleavage reaction that's associated with this as well. That has yet to be worked out.
So for vaccinia, the termination occurs, and then the 3' end is just polyadenylated.
Yes, that's a fair statement at this point. Yeah, certainly that's true for early genes. It's not quite worked out for. There's certainly one, but probably 4 different late genes where, in fact, you make a precursor that is cleaved by a viral protein and then polyadenylated. It's not known whether that's a global reaction or that those guys are just special.
Okay. Now, from the cell's perspective, with cellular polyadenylation, what purpose does it serve to have the polymerase continue past the poly A signal and continue transcribing, and then you've got this tail that has to be lopped off before the poly A is put on?
Is that a why question?
Yeah, he disguised it.
I just slipped it in there, didn't I?
I don't know the answer.
I don't know the answer to that. That's bureaucracy talking. It worked.
Yeah. It doesn't. Yeah, it doesn't need to be changed. Is possibly the reason.
It worked.
There could be a reason, or it could just work this
way. Well,
way.
actually, cleavage and polyadenylation is a regulatable reaction.
Sure.
And as a matter of fact, it's part of the whole splicing thing in a way, because if you don't cleave and polyadenylate, then you can have more RNA down the road. So in terms of this sort of splicing reaction and modular use of exons, if the polymerase just goes on until it finally terminates, you can regulate how many exons downstream you might transcribe by deciding whether or not you're going to actually cleave. And there's evidence that cleavage reaction actually influences whether or not the polymerase terminates downstream. So that if you cleave, it terminates. If you don't, it keeps on going. All right? Which makes sense, because if you decide that you want to incorporate a few exons downstream, you can repress that cleavage reaction and the polymerase will keep going. So there's probably a reason for that.
Right, and with differentially spliced genes, you would obviously you'd have sometimes you'd want to do it one way, sometimes you want to do it another.
Mm-hmm. Mm-hmm. Yeah. And partly you can do that by regulating that cleavage and polyadenylation reaction.
Right.
And one thing we didn't mention in this is that the cellular RNA Pol2 has what's called a C-terminal domain, which is this big repeated stretch of amino acids that hangs off one of the large subunits that is subject to regulation by phosphorylation, and depending on its phosphorylation state can bind a whole bunch of other factors, and so this thing can alter the behavior of the RNA polymerase as it's transcribing DNA and influence, for example, whether or not the polyadenylation cleavage reaction is going to happen. So this thing can be regulated in transit. okay, through modification of the C-terminal domain and the consequences of that reaction.
And when it does stall, the polymerase, if you remember from the last TWiV, if this happens to be a B cell, and that's an immunoglobulin gene being transcribed, that's when somatic hypermutation is thought to occur.
You know, it's been known for decades now that polymerase doesn't transcribe at a uniform rate, that it pauses, and there's a lot more consequence to that than anybody has anticipated before.
Yes.
There's a lot of genes that, in fact, are in the cell pre-initiated. The heat shock genes, for example, they're pre-initiated, and the polymerase sits there downstream from the initiation site, paused, so that if something happens, heat shock, some sort of stress response, it doesn't have to recruit everybody to the promoter and waste all the time doing that. They're ready to go.
Right. It's like keeping the B-52 parked on the runway.
Exactly. Right. So all of that, the regulation of elongation and regulation of those pausing steps and stuff is all critically important.
All right. That's transcription. And if you have questions, you know where to send them. TWiV.tv. And speaking of questions and comments, let's read a couple. We have a comment this is from Twitter. Just Another Kinase is his name, or her name. Dear Twivitrons. That's a great one. Twivitrons.
I like that.
For your information, mock is a UK term for a practice exam. Loving the virology on TWiV 161. Please keep it up. So that was mocks, right, from last time.
Okay.
There you go. It's a practice exam. Alan, would you care to read the next one?
Right. So this is from Gary. So, dear wonderful TWiV people. I just finished listening to your 161st episode, well-named Concerto in B. Really well done. I felt guilty that as a 30-plus year practicing pediatrician, receiving my undergraduate medical school education when most of this was necessarily only vaguely guessed at, I have not had the time and energy to explore the literature and bring myself up to date. This last podcast, along with your previous work, has provided a truckload of information that has made what would have been a K2 climb into an easy and fun one. Please consider dedicating a few of your future programs to more exploration of immune function basic science. In any case, I love listening to you guys. And Gary is a doctor out in California. So thank you very much.
In a nice area of California. Beautiful.
I think we ought to do a little more immunology.
Absolutely. You know, I learned so much from that.
That's great.
Was talking to my immunology buddy today and telling him about the last podcast we did, that Concerto in B, and he was not familiar with that particular paper, and he couldn't believe the stuff that was coming out of my mouth. I was actually educating this guy. It was great.
Cool. Yeah, it was great. Gabriel did a great job.
He did. Yeah. It was fantastic.
We got a lot of nice comments about that.
Even though it was 2 hours long.
Yeah.
Yes. Well, when we have a great guest who's explaining something that's that important, it's keeps people's attention.
And it's nice to have all that together, actually, in that one episode. We did a lot of stuff.
Yes.
Rich, can you take the next one, please?
Sure. Thomas writes, Dear Twivers, firstly, my sincere gratitude for all the work you put into making TWiV an absolute gem of a podcast and a medium through which I can still feel involved in basic science and virology, even though my studies currently have me memorizing various proteins and associations of the coagulation cascade in medical school.
I am so sorry.
Yeah, me too. In particular, memorizing.
Yes.
You know? What a drag.
And just for encouragement, you will never need to know that.
Unless he's a hematologist, right?
With that run-on sentence out of the way, I would like to comment on last week's TWiV regarding the SIDRAP release, saying that I think you all handled it very well. This was the bit where there's been a lot of press about the NSABB considering whether or not to publish this...
the flu virus that's going to kill us all.
The flu virus that's going to kill us all, right. Perhaps I make this conclusion due to my opinion being that which is shared by you all. Nonetheless, to suggest that science has a role in the progression of bioterrorism is probably not an incorrect statement. However, to suggest that scientists should refrain from certain works as a result of fear that knowledge and advancements may fall into the wrong scientists' hands is ludicrous, and I'm glad you addressed this release. This brings me to the reason I write to you all today. I recently came across a video that shows a one-on-one discussion between an out-of-character Stephen Colbert and the always fascinating Neil deGrasse Tyson. Briefly, the two discuss various issues regarding science, society, the universe, and knowledge. The entire discussion is well worth the 1 hour and 24 minutes. However, there is a short section which I hope for you and all TWiV fans to watch, which I think best portrays my feelings, and I'm sure it's many others' feelings, regarding the issue that you discussed about scientific discovery in fields that are implicated in potential biothreats. The section runs from about 16 to 1740 minutes and ends with Dr. Tyson stating that, quote, everyone blames the scientists and we are collectively part of a society that is using or not using to its benefit or its detriment the discoveries made by science. At the end of the day, it's not the discovery itself that is immoral, it's the application of it. End quote. They go on to discuss many other issues I think are important regarding the scientific community and what role we take in society now, but I particularly like this one quote because it reminds us of an ever-developing collective scientific knowledge, but does not blame the immoral application of this knowledge on science itself. Either way, I hope you all enjoy the video, keep up the great work, and again, thank you all for the weekly edutainment. Sincerely, Thomas. So that I have not looked at this, but that sounds terrific.
Yeah, I love Neil deGrasse Tyson. He's a lot of his comments are just spot on.
Well, thank you for that, Thomas.
I will have a look at that.
That's great. The next one is from our friend Kathy Spindler. In TWiV 145, the Invincible TWiV, a listener, David, wrote in to get suggestions of viruses that alter host behavior, resulting in increased transmission of the disease. He cited some parasitology examples in the classic rabies story, where rabies induces aggression that causes the infected animal to bite and transmit the virus. In TWiV 150, you read a suggestion from another listener, Kimberly, who described how baculovirus-infected caterpillars climb as high as possible, and then when the caterpillar melts from the infection, all the virus goo can fall down onto lower leaves. I have another suggestion, and that is viruses that cause pox, P-O-C-K-S, including both varicella zoster virus, a virus in the herpes virus family that causes chicken pox, and Rich's own vaccinia virus. In both of these cases, the pox or pustules that form are full of virus. The host response to infection causes these to itch, and scratching of the pox then can release and spread the virus. I haven't been able to find a good explanation of why mast cells or basophils might release histamine in the vicinity of pox. Maybe there's another explanation for the itchiness. Another case might be any of the respiratory viruses that result in coughing or sneezing. Think of the famous sneezing images, such as the top hits in a Google image search for sneeze. Cheers, Kathy.
Those all make sense to me. I like that.
Yeah, if you go beyond, overt behavioral alteration, then sneezing and scratching make perfect sense. Yeah.
Right. Physiological. I would think of these as physiological alterations in the host that aid infection. And if we're going to that category, you could go with viruses that cause gastrointestinal problems. Mm-hmm. Because these are usually spread by poorly treated sewage or poor hand hygiene, and if you're causing projectile vomiting or severe diarrhea, then that's a good way to spread as a virus of that sort.
All right, shall we do the last one?
Sure.
Alan.
So Aisha writes a few questions on HPV. Maybe you would be so kind as to pose a few questions to the next HPV expert you have as a guest. There are menstrual tampon alternatives that women can reuse called the Keeper trademark and the Moon Cup. I was just wondering whether women using these products who are infected with the high-risk HPVs could or would shed virus, and if the virus could persist in these reusable cups and act as a place where the virus can persist and reinfect cells in the cervix, leading to more risk for transformation. I can't find studies addressing this question in PubMed. I wonder what kills HPV. Boiling, detergents, vinegar? How well does it persist in water? Maybe a cleaner could be devised to make these eco products safer if in fact HPV does persist in them. Maybe I should just send off an email to the HPV researcher you had on recently. I work on HIV-1, epithelium, and Candida albicans. I was really tantalized by the idea that retroviruses use the immune tolerance caused by gut flora as an opportunity to evade the immune system. David Moyes, a postdoc in the same group, has described a mechanism by which oral epithelium tolerates resident yeast, but sets off a defensive signaling cascade when hyphae become invasive. Many thanks for your awesome podcast. It inspires me every time I listen, and I recommend it to anyone who will listen. Thank you very much, Ayesha. And in fact, we did fire off an email, it looks like. We have a reply from Michelle Osbun, who says, really good question. HPVs as non-enveloped viruses are much more resistant to desiccation and environmental assaults compared to enveloped viruses, such as HIV or flu or herpes. My friend and colleague Richard Roden showed some time ago that HPVs are sensitive to 70% ethanol. This is what we use to disinfect in my lab when working with papillomaviruses. Richard's work also suggested that boiling for 1 hour would reduce infectivity to near baseline.
I hope so.
Yeah, I would certainly hope so. My lab reported that the particles are sensitive to acid and probably will fall apart in undiluted apple cider vinegar, pH typically between 4.25 to 5.0. I recommend that the products be soaked in ethanol or 70% isopropanol, that's rubbing alcohol, for 1 hour or boiled for 1 hour, depending upon the nature of the product, before thorough rinsing in water. And she cites a couple of papers. I'm sure that both women and men shed virus as a route of transmission. However, I don't think anyone has investigated the reinfection issues posed, and little is understood about reinfection or infection of adjacent tissues in an infected individual, though I suspect this occurs.
There you go.
So there you go.
Good answer.
Thanks.
Good question. Good answer.
Thanks,
yeah.
Thanks, Michelle. Which brings us to our picks of the week. Alan.
My pick of the week is just a fun one for the season. I don't know, Vincent, I don't know if you're familiar with the National Weather Service radio that you can tune to. I'm sure Rich is, as a sailor. So they, NOAA, National Oceanic and Atmospheric Administration, the National Weather Service is under them, and they run a radio station that transmits in the VHF band. So if you have a radio on your marine radio for communication on your boat, it usually receives NOAA weather radio as well. And several years ago, they went to using a synthetic voice for this. The voice that they usually use is one named Paul. This is a computerized voice that reads the weather. It's much more efficient than the old having somebody have to sit down at NOAA and read the forecast. And the video that I've linked to is a happy holiday wish from the robotic voice at NOAA Weather Radio. It's a YouTube video, and it's Paul singing Deck the Halls.
Cool.
Great.
Very nice.
I had a lot of fun with that.
Great. Perfect timing. Thank you. Rich.
So I've been anticipating doing this for a while, but I had to do some revisions. This is a website called the Vaccinia Virion 3D Tour, and it's something that I put together with my colleagues Nassim Musachi and Paula Trackman. Some time ago, we wrote a review on Vaccinia structure and assembly, and in the process of studying for that review, it became apparent that there were really no good three-dimensional reconstructions of the Vaccinia Virion, and a lot of the thinking about what it looked like was, in fact, not correct. And so we sort of reviewed all the electron micrographs and stuff and put together our own three-dimensional model. And we wanted to be, it's hard to envision in 2 dimensions on paper, so we wanted to put together a genuine three-dimensional model. It turned out that Nassim's son, Michel, is an industrial designer. And so he has facility with software where they can build models of this or that, and then rotate them, mess around with them. And so we have this website that has several tabs on it that just show the model and different sections and stuff, but there's one called 3D, and unfortunately, this only works with Internet Explorer on a PC platform. But I think if you have a Mac, you can run the Mac in PC mode and run Explorer on that, and it will work. And if you go into the 3D tab, it'll download a little piece of software called eDrawing software on your computer, and then you'll have this model of the virion, and it comes in several components, and you can make various components transparent or remove them, and then you can rotate it around, and you can look at several different sections and stuff and get a look at what this really looks like. So,
and I will point out, if you're on a Mac and you don't want to do the whole Windows thing, if you click on the Images tab, you get a little animation showing different sections of this that's actually quite illustrative, I think.
Mm-hmm. Mm-hmm. It's nice.
So there you go.
So how did you end up making it for IE only? Was that you?
No, that wasn't me. That was Michelle. Okay, Nassine's son.
Okay, got it.
That's all the software will do.
Got it.
Okay, so it's the software's problem.
Okay, thank you.
And I have
Rich.
to say, doing it in brown like that, it really does look kind of like a viral moon pie.
We struggled with the colors. Let me tell you, we had some other colors that we messed with, but that's what we came up with.
I've known about this for a while. I'm glad you finally fixed it up, and
yeah, it looks good.
Looks great. People should enjoy that. My pick is a story about a 17-year-old high school girl who won a $100,000 Siemens Prize for making anti-tumor nanoparticles. Now, this is actually a story that my younger son, showed to me. He, once every 2 weeks for his science class, they have to find a science article and write a summary of it on a blog post. So he had picked this one, and I was looking at it, and I thought, this is pretty cool. So there is a site at George Washington University that I've linked to, which explains the story. And Angela designed nanoparticles that contain an anti-tumor drug which would be released in the tumor when you irradiate the tumor with a laser. So she is a junior in high school, and she wants to be a scientific researcher. So pretty cool and advanced stuff for high school, but it's great that at this level she's doing really neat stuff.
Yeah.
I thought that was pretty inspiring. And that will do it. Oh, no, we have one more thing. We have a listener pick.
This is actually a bundled pick. This is about 3 or 4 picks in one, I think. But
yeah, this is from Sarah. Hello to the TWiV crew. Here are a couple of picks I thought would be good for provoking thought and generating discussion. While working on a project, I came across some papers attempting to define what critical thinking means. I appreciated the following quote, and it reminded me of the many instances in TWiV where you hosts have discussed the scientific method and thinking critically about research findings. It came from a consensus statement on defining and promoting critical thinking, published in 1990 by a university dean. Quote, the ideal critical thinker is habitually inquisitive, well-informed, trustful of reason, open-minded, flexible, fair-minded in evaluation, honest in facing personal biases, prudent in making judgments, willing to reconsider, clear about issues, orderly in complex matters, diligent in seeking relevant information, reasonable in the selection of criteria, focused in inquiry, and persistent in seeking results which are as precise as the subject and the circumstances of inquiry permit. End quote. And that rules me out.
I was going to say that describes everybody in Congress, doesn't it?
Didn't they forget trusty, thrifty, and brave?
That's right. Boy Scouts. Wow, that's pretty tough.
Yeah, that's tough.
I don't think there's anyone in the world that meets all those criteria. Do you?
Well,
it starts off the ideal.
Yeah, right.
If you have maybe a few of those, you're okay.
Yeah.
Okay, so she gives a link for that quote. Next, perhaps less of a pick and more to stimulate episode topic ideas for those of us who are into the medical side of virology. Here is a paper about diagnosing viral acute respiratory infections using peripheral blood gene expression signatures. If this seems interesting to y'all, Dr. Zoss and or Woods would be great guests on the podcast. They're both infectious disease physician researchers. She sends a link to a paper, Gene Expression Signatures Diagnose Influenza and Other Symptomatic Respiratory Viral Infections in Humans, from Cell Host and Microbe. Last, Dr. Woods would be an interesting guest for another reason, his interest and teaching activities in a movement called One Health, which looks at human health from a human, animal, and environmental perspective. I'm still wrapping my head around exactly what One Health is, but apparently it's even caught on at the CDC, and she sends a few links for that. Keep up the great work and keep having fun. Thank you. We are having fun.
So she ripped off my One Health pick. That was on my pick list.
Really?
There's a virologist of some repute, Tom Monath, who was a prime mover in putting that together. And my understanding is that the most important component about that is the recognition that the majority of really nasty human diseases and emerging human viral diseases or infectious diseases are of zoonotic origin. And so it's important to have the veterinarians talking and the people in agriculture talking to the doctors, and vice versa, probably more important, vice versa, the doctors talking to them, so that we can keep an eye towards the future and survey some of these things. And so that's a lot of what One Health is about, to sort of take a more global perspective on, in particular, infectious disease, but other diseases as well.
Oh, that's cool.
Could be an interesting guest, right?
Who, Tom Monath?
Woods.
Or Woods, yes, absolutely.
Yeah,
Either one.
We'll put
but Woods, yes.
So thank you, Sarah. We'll put it on the list. And that will do it for Virology 101 transcription.
Bingo.
Yeah.
All right.
You can find us, of course, at iTunes, at the Zune Marketplace, at microbeworld.org. You can stream episodes to your Android or iPhone with the Microbe World app at microbeworld.org/app. And if you are new to the podcast, please go over to iTunes and leave a comment. It helps us to stay more visible in the iTunes podcast directory. And, of course, send your questions and comments to TWiV at twiv.tv. Alan Dove is at alandove.com. Thank you, Alan.
Always a pleasure.
I hope you learned something from
I certainly did.
This episode.
It sounds to me like he was pretty much up to speed from the get-go.
Yeah.
It was a good review, though.
Excellent. Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
Absolutely. Great time as always.
And I'm Vincent Racaniello. You can find me at virology.ws. I always enjoy the Virology 101s.
Yeah, they're good. We need to do them with a higher frequency.
Yes.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
'68 was probably, I saw the Doors play in the Fillmore in San Francisco. I saw the Jefferson Airplane play on the boardwalk in Santa Cruz.
Cool. That's neat.
That was all around that time.
Listen, speaking of the Doors, you know the famous Jim Morrison quote.
No, what's that?
Love hides in molecular structures.
He said that?
Bert Semler has that at the end of his email, yeah.
That's pretty good. Good for him.
We got to use that for an episode title sometime.
Good for him.
Isn't that cool?
Yeah. And we could have the image could be Jim Morrison.
Yeah, I just found that's lyrics. It's actually in one of his songs.
Really? Which song is it?
Love Hides.
Love Hides. You think he knew what a molecular structure?
Yeah.
Do you think
he probably saw the molecular basis of life while he was molecularly stimulated.
Mm-hmm.
Whatever.
Yeah.